Okay, good, and thank you everyone. Thank you, Rajiv, and all the organizers for the really nice invitation to come. And I'm glad I could actually make it finally this year. So I'll just talk briefly about all of what we've been doing, some of the work we've been doing with GWAS, and just all the different ways we can apply this in the breeding program, and even extending this out to like seed certification and stuff like that. So just by way of introduction, if we think about the breeding process, we have a lot of material, elite material, some unadapted, some exotic, and it goes through this really like funnel-like process. And this is many years, a lot of resources typically go into this type of process. And so we're always thinking about ways to make this more efficient and larger and faster using molecular information. And so things like optimizing parent selection, optimize diversity and crossing, F2 enrichment type like marker-assisted selection approaches, genomic selection for whole genome when you get to these more difficult traits like yield, and then things like marker-assisted back-cross conversion. And in the end, we produce varieties, and we want to make this go faster and more efficient. And so just to really oversimplify, but to think about the types of markers that we want and the objectives, you can think about single locus typing or a whole genome profile. And so this is typically targeting known genes that we know have a large effect, and we want to drive the allele frequencies in the breeding program. On the other hand, this is actually the whole genome, typically to look at population structures and relationships across all the material. And so just different types of applications for these. These are genomic selection and association mapping approaches. But the big thing when you're a breeder, you think about cost. And so here you typically think about cost per data point. Here I tend to think more about cost per sample. So once you reach thousands or tens of thousands of markers, at least from a genomic selection perspective, additional markers are not that valuable relative to lowering the per sample cost, which increases your population size, and you get a lot bigger gains that way. So this was the trend line shown earlier, and yeah, the sad thing here is it's kind of leveled out for 2 years. But thinking about genotyping by sequencing and GWAS, I always show this slide, and this is really driven by these amazing developments in sequencing. It turns out this is really good for wheat and other polyploids, where we have context sequence now rather than just assaying a single point mutation. And then I think the big one, especially for diversity studies, but also for germ breeding and germplasm, is that we discover all the polymorphisms simultaneously. And the biggest one here again for breeding programs is that the per sample cost is really low. And so the GWAS approach that we take uses a reduced representation here, and this is restriction enzyme based. And so we primarily use a 2-enzyme combination, PST1 and MSP1. And they're inexpensive because we put a lot of samples together, and then the sequencing cost gets spread out across all of those samples. And so, still by way of overview, just to really simplify this whole process, going from genomic DNA to making libraries and then sequencing those, and then really streamlined bioinformatics process. And here again, this is so we can move through a lot of material, make selections, and then discard the ones that are not selected. So now I'm just going to go through a bunch of different applications of how we've been using GWAS. The first driver, first application we're mostly interested in was in genomic selection. And so just highlight some of that. So the keys for genomic selection, here again, this is probably familiar, but starting and needing a training population basically to train the model, and then that's applied on selection candidates. And so in the flow of this, and so the real key here is because we're discarding 50 or up to 90% of the material after its genotype, is that we want it to be inexpensive. I just like to throw in here also, though, that we need accurate phenotypes. And so the upper bound of our prediction models is always the heritability of the trait. And so if you have lousy phenotypic data, then you're going to get lousy predictions. And so this was our first study where we used GWAS for genomic selection, and it was really just this question: is it a suitable marker platform for GS? And so this was on some cement material from the Semi-Arid program. We had a pretty small set of lines, but enough. And the key here was that these were in replicated field trials from Oregon, so really high quality phenotypic data from these good replicated field trials. And so we genotyped these with the typical GWAS, generated about 35,000 markers after filtering, and then just applied a simple ridge regression. And so you can see here we were able to fairly well on this PCA, first and second PCA, discern some of the population structure. These are colored here by families. And then we obtained prediction accuracies for yield. This is grain yield under drought and grain yield under irrigated conditions. Prediction accuracies of 0.3 to 0.4, up to 0.5 in some of the trials. And so this was for a low heritability complex trait. This was pretty encouraging and good prediction accuracies on this first study. So to look at other traits, and here again for genomic selection, the real payoff is the more expensive and the more difficult that the phenotype becomes, the more valuable it is to get prediction with the genomic selection. And so a great one in wheat is obviously wheat quality, where we think about milling and baking quality parameters. And so this again, this study I highlight here was on cement bread wheat lines, and these again were from replicated yield testing. And so we ran GWAS on the same set of material, and then used just a cross-validation approach to get an idea of model prediction accuracy. And so this is a really busy slide here, but what I want to point out here is for all of these different quality parameters, including test rate, grain hardness, mixing times, loaf volume, had really good prediction accuracies in the range of 0.3 up to 0.6 and 0.7. And so, and just an example of two of those, this is a mixograph, and so you're actually milling the flour, and then you mix the dough and you look at the dough strength. And so these traits are measured in minutes. And so obviously not real high throughput, but we had really good prediction accuracy. Same thing with a loaf volume, so actually baking a small loaf of bread and then measuring the volume on that. So obviously here again, a low throughput expensive trait. And so if we can predict these kind of traits with any type of reasonable accuracy, I think this is really where there's going to be a huge advantage to applying genomic selection. The last study to highlight here on genomic selection, this is some stuff from the hard winter wheat region in the central U.S. And this is what we call regional performance nurseries. So each of the breeders each year will contribute 5 to 8 entries, and then the total set will be about 30 entries. And so this is really analogous to the international nurseries that are coming from CIMMYT. These sets of lines then are sent back out to all of the breeders and then grown at 40 to 50 locations each year. And so we have a lot of historic data. These were established in 1931. For this set, we went back to 1992. We genotyped all of these entries, and then we used the historic data on tens of thousands of plots and hundreds of thousands of observations. And so this set, for just the southern sets of nurseries, is about 800 entries. And so now what we can do is we can really look at predicting across locations, across years. And if you're familiar with the climate in this region, it's really highly variable from year to year and location. But what we came out with was here, this is on the bottom here, showing the prediction accuracies basically across the full set of material across all locations. And what we can do is, because these are breeding nurseries and oftentimes a few of the entries are repeated year after year, so you can really get a baseline, which is what I'm showing here, a baseline of the phenotypic correlation from one year to the next. Okay, so that comes in at about 0.28, I think. And so for each one of these points here, you can see that 9 times out of 10, the genomic selection prediction is as good or much better than what we can actually obtain with the phenotype. And this here again is because the environments are so variable, and because also with genomic selection, we're leveraging the information across many years of evaluations to start to iron out those differences across environments. What then we can start to look at again is to start to look at genetic correlations across these environments. And so here's all of the locations shown with these points here, and then these lines and the colors, the width of the line, and then the color are showing the number of years and the strength of the correlations across these locations. And so we can see, and this correlates well with breeder knowledge of the location, that a lot of these locations have very good correlation year to year. And then when we get to these drier, more extreme condition environments, the correlation really goes down year to year and location to location. So this also can really start to inform how we do multi-location testing and dividing sets of entries across locations. And so just where we're at on applying the genomic selection and some of the projects we have going on, this is a new project that we just kicked off over the last couple of months in partnership with Simit and Cornell. And this is applying on a large-scale genomic selection in the Simit breadfruit program. And so on this flow diagram, really looking at the generations of material coming through the program at Simit, Mexico. And then what we're looking at is taking and phenotyping these same entries across 4 locations in South Asia, 3 locations in North India, and a site in Pakistan, targeted specifically for optimizing yield under heat stress conditions. And so that's what we're showing here is just the different locations, and here they are on the map. And so what we'll be doing is there's about 1,000 entries that are going to each of these locations each year. The previous set of material, about 8,000 to 10,000 entries, we're genotyping in the Simit program, and then at that point we'll be developing these models and applying the genomic prediction and selections at that point here as well. So now I'm going to move on to some other ways we've been applying the GWAS. Here again, just to kind of highlight it's a pretty versatile, flexible tool for different types of approaches. And so now just to think about characterizing genetic diversity. And so a little bit of background on this, thinking about in wheat, we're talking here, going from wild species to early domesticates, there's a bottleneck there. And then in the intense modern breeding process, there's been further bottleneck of these populations. And so what this looked like in wheat, I think Dave gave a really great overview here. But wheat is hexaploid wheat that we call bread wheat, went through 2 rounds of whole genome hybridization. First to produce the tetraploid, and then with that hybridization, the Taschii to produce the hexaploid. And this hybridization event here, going from tetraploid to hexaploid, was an extreme bottleneck in the wheat bee genome. And so what we can do is we can go and compare that to some of these rich genetic collections that we have. And so at Kansas State in the Wheat Genetics Resource Center, we have 500 some unique accessions of the Taschii species. We genotype this whole collection with GWAS, and just to also note that I'll show how these are physiologically classified as well. And so here's the distribution and the location collections of those accessions across Central Asia. And here's a nice picture of this kind of, it's a weedy species. And so what I'm showing here is the genotypes for all of these collections put onto the first and second PCA. And so you can see right away that there's pretty strong genetic differentiation into 2 previously known 2 subspecies, and then from that differentiation here. Point out that this PCA explains like 56% of the variance. What I'm showing in colors here is actually the physiological classification. And so you can see there's the different, what they call subspecies but there's actually pretty poor classification. And this wasn't a real big surprise because it turns out the morphological characteristics aren't actually that good at describing the different subspecies. What we're showing here now is the third and fourth PCA, and these are colored by location of collection. And so now we can also really see that we can do a good job also differentiate geographic origins are evidenced by the genetic separation. And so what we want to do then is we want to put this back into the context of wheat, the hexaploid genome, and the breeding material. And so now colored in green is the Taschii germplasm collection, and then wheat here is about 600 entries from the Summit breeding program. And so you can see here on the first and second PCA and the third and fourth that, as is well known, that there's very limited diversity relative to the wild species. And so now to think about how can we take really informed approaches to integrate this diversity back into the breeding program. And so that's what we see here in the breeding program. So now I'll move on and talk about some genetic mapping, and this is GWAS turns into be a really easy, inexpensive way to develop some high-quality genetic maps. One example here, this is on the synthetical product population, and showing here mapping stem rust resistance to 2 races of stem rust in this population. And you can see you can generate an extremely high-density genetic map, and then from that you can really start to map with high-precision QTLs and genes across this population. And so here again, it really makes any type of mapping study very tractable and relatively inexpensive. The cool thing is that because, like I said earlier, we can discover SNPs at the same time we do the genotyping, this really enables moving into these wild species, moving into basically any species we want, and straight away developing some really nice genetic resources. And so this is an example from Phenopyrum intermedium. It's a wild perennial grass species that has some potential for domestication as a perennial grain crop. And here's only showing one set of the linkage groups. It's a genome pretty similar to wheat. It's 15, 16 gigabase genome, hexaploid, allohexaploid, but it's also outcrossing. So we developed an F2-like population from just one self individual and can put together a really nice high-density genetic map. And so the key here is then we can really think about having tractable genetic resources for basically any species. And so what we can do then is we can take this information back to the breeding program and immediately start to apply genomic assisted breeding approaches. And here again, you can kind of do away then with this idea of orphan crops. At the same time, we have whole translocations and integressions from intermedium into wheat that harbor valuable disease, virus, insect resistance genes. And so this genetic resource then gives us high-density markers on those chromosome arms to start doing chromosome engineering, fine map those genes, and then move them into small segments on integressed into wheat. So now I'll talk about some marker-assisted selection, and then this gets into that framework of having single markers relative to a whole genome profile. And so one thing we've been like trying to work out now is what we call spiked GWAS. And so the idea here was to have an open, flexible platform where we can target single loci at known targets, and do this basically independently of our whole genome profiling. And the real key here is that we just use a really small percent, a small capacity of the total sequencing lane, here again to make it cost effective, because and you're doing a few number of targets, you don't need a ton of sequences. So that makes it cost effective, makes it high throughput, and it makes it flexible because now what we do is we use a single set of barcodes that are different from the sets of barcodes used for GWAS. And so kind of what the flow of this whole thing looks like is earlier I showed developing the GWAS libraries here. These are digested adapters, and then they're amplified and pooled into a single library here. At the same time, on a completely different set of material, we can amplify known targets, and then with the N13 tails, we can append on different sets of barcodes. We can pool all of those into a separate tube, quantify those, and then we just add them in at 1%. And so what happens here is we can separate these back out after sequencing. On this side, we can generate something like 50,000 markers on 96 to 192 individuals. They're really low coverage, just like at 1.5 to 2x coverage at most. On the other side, using 1% of that, you have about 2 million reads. But if you're doing something like a target of 10 markers on 384 individuals, you can have like 500x coverage on those. And so you can really start to eliminate missing data. At the same time, it's really high throughput. So we tested this out on some heat genotyping. We made a typical GWAS library, then we amplified 4 SNP loci separately and added those in at 1%. One thing we tried was we just converted Caspar markers simply by taking the selective base off of the end, and so that now rather than doing a competitive PCR, you're just sequencing the known SNP. And so that worked really nicely. What I'm showing here was one of the Caspar markers where you can see this is the number of reads. The read count, each one of these points being an individual. The read count for allele A and the read count for allele B. And so you can easily classify those, a few heterozygous here. These were more as dominant markers because we're picking up the same paralogs on other genomes, but at the same time you can easily classify these markers based on the read count for individuals. So now the final thing is just to talk about how we can even tie this into what the seed certification process. And so now we're just starting to routinely apply this technology into checking certified seed lots to make sure that they're the correct variety. And so we talk a lot about breeder seed and kind of in the breeding process, but then there's all these important steps that this goes to foundation seed, it gets increased, and then it gets grown and produced on farms. And so at this step, there's opportunity, and through the certification process and through going through many different channels, there's opportunities for varieties to get switched, and a farmer could be receiving seed that is something that they did not order. It could be a variety that's unadapted or a variety that's not well suited, that's just mislabeled. And so what we're doing in the Kansas Foundation seed is we're starting to implement genotyping of every certified seed lot that comes through, and then through that we can check the varieties. And so here's just a nice little case study on that thing. And this was from one of the extension offices. There was a farmer that came in and said, hey, I purchased this seed. It doesn't look like what I thought it was. And so they sent us just these 2 samples. We genotyped them and checked them against each other. They were less than 5% heterozygous, so that told us that they weren't like seed mixtures, that they were pure line, truly varieties, and they were only 74% identical at all the shared markers that were not missing data. And so that told us that, so really for any 2 random varieties out there, they're anywhere between 60 to 80% identity. And so we could tell that they're different varieties pretty easy. We have a huge set of reference panel that's been genotyped just as part of our routine breeding efforts. And so we could go in there really easily and say for sample 1, it was 98.7 identical to the variety Fuller, and the other one was 99 90+% identical to endurance, and these are well within our 1% error rate for GWAS. And so we can easily go in, check the variety, and then give that information back to the farmers and the seed growers. So with that, just want to give all the acknowledgments. My research group, Shuangye Yu, really runs the GWAS pipeline, generating most of the material. Trevor Reif has done a lot of the genomic selection modeling that I talked about today. And Tracy has worked on the intermediate wheatgrass stuff. Also the wheat genetics resource group, my group, along with the Bickham Gills group. And Alan Fritz runs the wheat breeding program at Kansas State. The group at Cornell is instrumental in all of the initial stuff we did with genomic selection. Jeff Edelman, now at Wisconsin, worked with us a lot on the genomic selection stuff. And then, of course, the group at Summit, which does all the amazing breeding work. Ravi and Suzanne, especially, are key in these big genomic selection projects. So with that, I'll stop. But I do need to acknowledge USAID, Gates Foundation, and the Wheat Alliance are major funders of all the work we're talking about.
But in this case, I think there cannot be a dispute. I am sure all of you will agree. The father of the diversity array technology, Andrzej.
You can hear me, yes? I can raise my voice or raise the mic. I'll try to both. Both methods. How about now? Okay. So thank organizers not only for inviting me again to ICUSAT. I always enjoy those trips, but also for putting me at the end of this session and after 3 very interesting talks that put a lot of groundwork for what I'm going to say. You know, I really want to make some very brief introductions and then talk briefly about genome profiling technologies that our organization has developed and delivers, but also a bit of data analysis component. I'll try to focus on data storage and management platform that we have developed and brief conclusions. So I'm recycling some slides and providing just updates. So for those who saw something similar recently, I apologize, but there are probably some people in the audience who never heard about us. So the diversity arrays technology is a 13-year-old child, baby. I was stimulated by the first presentation of someone saying that he feels old at the age of 38. I have to say that I'm nearly 20 years older and I should feel half dead. I still think that I'll be defying this tendency to consider someone approaching 60 as half dead. I think that we have still plenty of juice. So those all these sticking. So coming back to our company, compared to myself, it's still very young, and we're mostly doing genome profiling services and technology development, but also increasingly IT support for agriculture. At the moment, we are a team of 24. We have like 3 different legs, so we like tripod. We partly overlapping with people both in the laboratory, then in data analysis, and also sort of straight IT team. And on average, we have about 5 international visitors in the company at any time. We are global service providers. In the last 14 years, we process about 700,000 DNA samples for whole genome profiling. At the moment, we are at 150 different organisms, and every week we add 1 or 2. We are very far from BGI in terms of size of the number of organisms and the density of profiles, but we're also much smaller than the team at Shenzhen. So we mostly work in crops, and our initial work was really, apart from wheat, a lot of orphan crops that did not have any technology accessible to them. They relied on DArT platform for initial entry into genome profiling and the genomics. 95% of our business is still outside of Australia, and through training, we had probably close to 100 different visitors, most of them from lesser developed countries. But also in service provision, we are cheap enough to be accessible by organizations in lesser developed countries. But also we do a technology package transfer, including at some point to Intisar. So another ancient slide that I just wanted to make a couple of points. So we are quite young organization, but in terms of provision of the whole genome profile, we were actually the first to introduce technology that was able to work with practically any organism within a very short period of time. We were able to develop a profiling system that could compete with the array, SNP array platforms that were developed since then. And we spent a lot of time working with the technology to make sure that we are not captive of the ascertaining bias that was prevailing in the early SNP chip platforms. So this part of our technology development is in a way history. We stopped cloning libraries and developing new array systems. We're still producing data on arrays for those kinds to prepare these compared to DArTseg platform, but in a way with DArTseg platform, which is one of the genotyping by sequencing methods that were described by the speaker, especially by Jesse. I think that we don't have to worry too much about ascertaining bias any longer and take advantage of decreasing cost of DNA sequencing to produce data that detect not only SNPs but also insertion deletions. We can measure copy number variation, and also we detect methylation type of variation. So we started many years ago developing this DARTseg platform, and we started initially with developers of open access technology, the pollinator system, that unfortunately did not make a commercial success and was slowing down. And after some initial investment of effort and operating on this platform, we moved to Lumina platform, 2000 at the moment, that we read about 77 bases to extract our marker data. For third generation platform, we're really looking forward to dropping the cost of the cheapest sequencing method. Very importantly, when developing DARTseg platform, we made sure that it's a very generic platform that we can plug in any sequence data from any sequencer, whatever is the cheapest. So we're not bound to a particular sequencing platform provider. We spent a lot of time integrating these into a laboratory information management system and online ordering system. The DARTseg is also very tunable, as David said, that scheme GWAS is tunable. Same thing applies to DARTseg and GBZ in general. You can titrate the amount of sequence that you put into your analysis to extract marker density that is appropriate for the application that you have in hand. This technology is targeting, compared to some other GWAS approaches, targeting low copy, hypermethylated fraction of the genome. Therefore, it's predominantly extracting data from gene space. We use metric filtration, somewhat similar combination of enzymes that Jesse mentioned, but since we are working with 150 different organisms, in each case we do method optimization to make sure that we're providing the highest marker density for this given amount of money, so biggest bang for the buck, so to speak, for our clients. And after optimization, this particular method of complexity reduction is applied going forward. We sequence on average for different organisms, it varies, but it would be approximately 200,000 loci in the genome. And since most of our services is around 96 plex, then we have on average 10x sequencing, which brings corresponding data quality benefits in terms of high power rates and high level of reproducibility. So we're delivering both S&P information and also silico DArT. I stay away from presence-absence variation because, as David rightly said, presence-absence variation is presence or absence of a fragment in genomic DNA, not in representation. So there's a little bit of mess in the literature and how people use certain terminology. Just to be funny, we call them silico DARTs because those markers are actually in silico presence-absence variation analogous to what we were doing on microarrays in a traditional DART. So we also do a lot of downstream data processing, alignments to reference genome. We have it, but also mostly genetic mapping, dissection, and some other things, depending on the client's needs. So these will look very similar, so I'll go very quickly. I anticipated Jesse talking in some way. I'm glad that he covered a number of things that I wanted to mention here. So comprehensive germ plasmid characterization and diversity studies and guiding selection of parents, that's the best entry for any breeder to improve their breeding efficiency before any other technology that is listed before. So I strongly encourage those who have not done it in any crop. I think that by sequencing approaches give you a very easy entry into identity profiling that you can use for really understanding your germ crop. We do a lot of genetic ID, breeder rights work, a lot of it in horticulture, but also in other systems. And again, following what Jesse mentioned a moment ago, seed purity testing. About 4 years ago, we actually established this system in Australia with support from Grains R&D Corporation, initially for barley, then for wheat, and now we're doing it for many other cereals and also other grains. So we have in Australia selected in wheat over 200 different reference varieties for reference, and then we can check any sample going either from the field, from the farmers, or from silos, from the grain distributors. We provide these not only genetic identification, but we also develop a quite sophisticated way of quantifying impurity, so we can estimate in quite detail the level of impurity in those samples. And while it has been developed in Australia, as I said, several years ago and implemented quite intensely, this is the most recent picture that I added to this particular slide. It shows Ethiopian varieties of red wheat and durum wheat that we can use in one of the projects funded by the Foundation to learn more what is being grown in this country. We have similar projects in maize. We also do quite a bit of genetic and QPL mapping. We've done over 500 mapping populations just in wheat, and probably well over 1,000 other species. We do a lot of association analysis of GWAS. There were a number of examples that were shown, so I will limit what I'll show you later as examples to just a few selected applications. But for example, gene tagging and gene cloning is something that we can do quite inexpensively. Genomics, a lot of our high-density maps that we're constructing were used by organizations and social intelligence sequence assembly to help with those assemblies. We do quite a lot of genomic selection work for various organizations. Also, as of the last few months, quite dramatic increase in the the area of biodiversity and landscape genomics. And my background is in population genetics, so it's a very welcome addition to what we do. Just again mentioned mapping, so I'll go very quickly. Years back, we created 6,000 DArT array markers, and we actually identified a number of issues where we were trying to build a consensus for a species that is as diverse as possible. I think that it's important to understand that if you really want to have a genome sequence of wheat, you would have to do it probably many number of times, and then have de novo opportunity for any variety. Practically any map we do in a biparental population will show some structural variation. A lot of these structural variations are linked to disease resistance genes that are transferred by breeders into the cultivated material. More recently, we added integrated this initial map with map based on identified DArTseg markers. And an interesting point in map construction is that to change our mapping pipeline from medium density maps that we were doing on the ARI platform to those when we have every mapping population with about 20,000 severity markers. You have to change an algorithm how you handle that in order to produce a sensible map. So we spent some time developing this capacity. And our maps are not big and longer. They will look like density plots because you can display single point on the map any longer. And very brief example, it's a service for one of the European clients. They submitted, they wanted us to find some markers into a phenotype. When we perform something that we call quantitative bulk segment analysis, we find two sequence tracks that we can map to our wheat reference. It turned out it was transient, and you can see the probability value of 0 and very clean bulk with 0 signal indicate that we actually can identify very clearly 0 linkage disequilibrium and be within the gene, because those tags were transgene genes. So this was a very nice, almost scientific experiment that was designed by our client. So this gave us an opportunity to distribute this work to many other of our clients. With genomic selection, we again stepping after my speaker. So very briefly, all the work done on microarray platforms for Cornell University with some reasonable predictions. We've done some of our benchmarking for novel approaches to genomic selection predictions. We're developing in partnership with NICTA organization. We increased quite drastically the marker density, and we applied several algorithms both off the shelf, but also some of our new algorithms, and those predictions have not increased, certainly not drastically due to the increase in the marker density. So you have to be careful with the need for increasing the number of markers in organisms that have high linkage predictability. This is just an announcement of for the talk that Peter Wenzel will deliver on the FEED project. Very briefly, we're working with Semi-Arid their drone plasma collection both for wheat and maize. Maybe too fast. So we've done 50,000 wheat samples and 40,000 maize. But we're also helping them with IT support for marker data, integration with other type of data phenotypes in particular. We're helping with GWAS analysis and diversity analysis with the team, and we also transfer this technology to Mexico. I understand that Peter will mention something about it. This is just to tease you and wake you up. An example from animal studies that we've done just last month. I thought that no one will talk about that. I refer to this because I remember my talk about 10 years ago. The title of the talk, Size Does Not Matter. And it was with the reference to DART technology being able to work with any genome size and any ploidy. And I have to say that size does matter. For Gambusia fish, females prefer well-endowed male. And our project was on genotyping over 3,000 of these fish, finding parents, finding fathers. Mothers was quite clear, but finding fathers, and now we're still looking up what's the genetics behind this strange preference. Okay, so I hope that it was a welcome break from boredom of my talk. I have to tell you that it will probably make much more interest in general public than the crop improvement work that we are doing, which is a very sad statement, but anyway, I try to make it as light-hearted as I could. Now, we're moving from genotyping service to digital agriculture, and about 7 years ago, we recognized that a lot of our service provision is not yielding enough benefit for our clients who are not enabled to really use this kind of data efficiently. So we spent quite a bit of time developing new IT infrastructure for integration of molecular data, breeding records, field, but also environmental data. That resonates well with what our first speaker this morning said. And we also established collaboration with National ICT in Australia on data mining and machine learning. Also work with Semi-Arid and some other colleagues in Australia and also in Scotland. So in that context, we started working with statistical machine learning about 10 years ago. We thought that there's an opportunity to bypass some, for example, composite interval mapping approaches that were prone with problems and suboptimal algorithmic solutions for finding market-type associations and GWAS analysis, but we extended this machine learning technology to genomic selection. We also moved to NICTA to look into something that we can consider epistasis analysis. So we're looking not only in a linear model with one marker at a time, but also looking at pairs. So in the data set that will have million SNPs, and this is the data set that we're looking at from time to time, that gives you trillion possible pairs. And if you go to a triplet, interaction between 3 loci or 3 SNPs, that's quite mind-boggling search space to do. So we were very fortunate to work with people who have a technical solution to it. Their algorithms are about 2 to 3 orders of magnitude faster than anything else in this space. So we're trying to see whether we can improve prediction capacity deploying this kind of interaction between loci in our program. Now, moving to the KDDart infrastructure, IT infrastructure. As I said earlier, quite a few years ago, we realized that the uptake of DNA profile information is limited by inability to integrate this information with other types of data. So we set up a program of capturing this data from not only genetics but also field and data about environment. I think that this was viewed as a critical element of the system. Here is a pictorial view of how you have the field data, whether you use drones or some other more newer phenotyping, or you're working with the field with the tablet, or your combine is harvesting the yield. That doesn't really matter. These are all different kinds of phenotyping technologies that you can combine with DNA information, but also with GIS and also high resolution environmental data like weather stations that provide selective environmental characteristics. And the KDDart database or KDDart platform was supposed to bring this all together. I don't have, judging from the time, I don't have enough time to talk about it, but the architecture of the system is 3 layers. Database layer that in fact in our system has several different databases and more can be added. The critical element is data access layer, which is simply application programming interface. That's a glue that combines all the possible user interfaces that sit on top of the data access layer. And I'll just give you a very quick run through some of the interfaces. First, I'll say that user interfaces in KDDart system are designed to be recyclable and designed to be assembled in a way like Lego bricks. So we're building many of those partly overlapping interfaces, focusing on user interfaces that are web-based, but we're not eliminating options for desktop. Some of our user interfaces are actually desktop applications. And the important thing is that all these user interfaces have to be easily navigated by people with relatively limited IT skills. A few of the examples, actually 3D Explorer is developed by Ryan, is not only for users but also for software developers. Is a technology for any other programmer to get into the system, learn how the data access layer works, program new user interface on it. So that's a very useful application for the platform development going into the future. KDLog, our inventory management system, is an essential tool for operating bidding program. Inventory management is essential for efficient operation of any bidding program, and this technology was developed. Actually, we have two different user interfaces because inventory management is different meaning to different people. So we have C++ version and also phone grab version. Some of the screenshots on the phone grab on the right. 3D compute is a very important user interface for us because that's an analytical platform for all KDDart systems. It's generally a tool for integrating of any algorithm, even with limited technical expertise in IT. People can develop their own user interface for running their pet algorithm. Mindful of time, just wanted to say that there are a number of challenges. If you're in the business of developing software applications for agriculture, you'll be faced, especially in the breeding context, you'll face a number of challenges. And the biggest is the development of user requirement document. Capturing user requirements is something that is very difficult. That's obvious initial requirement for any proper IT project. However, capturing these user requirements as a breeder, especially in public institutions, proved to be very sorry. Let me finish now. Okay, so breeders in public institutions are overworked. They don't have enough time, and because there are often in larger institutions, several of them, we find it very hard for them to adopt a common vocabulary, common standards, and naming conventions. And I think that the management of pedigree is particularly challenging, but we're working with some of our partners, starting from very generic description of breeding cycles, understanding breeding use cases, and using these kind of business analysis models in order to develop software solutions that are long-lasting and efficient. Let me go to conclusion slides. So I think that I don't have too much time to give you examples, but they are sort of some are floating in literature showing that we have two quite efficient platforms, but obviously moving more and more towards sequencing platform applications. We're also delivering a complete technology package, meaning not only DNA profiling, but also a comprehensive data analysis and also data storage platform. We have done quite a bit of development in develop new tools for managing information and helping people to transit from traditional to modern breeding. We do it as a service, and we think that this is an important part of transiting of the breeding from the old days to new reality. The new methods for data analysis developed in partnership with our NICTA colleagues I mentioned them very briefly, but I think that KDDart data integration management platform is really a good opportunity for deploying these platforms for a number of public and private breeding programs. Important thing to remember is that communication is still the real challenge, and you know, being an older person than the first speaker or the speaker in the previous session, I would say that we get often overly excited about technology, not recognizing that the challenge is all the way, 95% of the challenge is in deployment. It's nice to draw a diagram of how things could work, but it's important to realize that you need to have business models. You have to have people with the right capacity. There's a lot of ingredients that have to be in place for the most fantastic sequencing technology or any IT tool to actually see the light of the day and have any impact on food farmers in India or globally. So I'll leave you with this thought. Okay, thank you.